Hi everyone, my name is Robert Huff. I am a first-year master's student at San Diego State University in the molecular biology department, and I work in Dr. Roland Volkovic's lab, and in our lab we study viral host interactions. I specifically study the mosquito-transmitted Zika virus that is associated with causing microcephaly in newborn infants. Now it's very important that I'm doing this research because there currently is not any antiviral treatment or vaccine options available. Now to give a brief overview of the virus life cycle, we have a Zika virion here, which will attach to a host cell by binding to a cell surface receptor, and it will release its positive sense single-stranded RNA genome into the cytoplasm. Now this genome mimics our messenger RNA that we have in our human cells, so that our host machinery will be able to bind to this genome, and it will produce a long viral polyprotein. Now within this viral polyprotein there is a very important enzyme, and this enzyme is called protease. And what this protein is responsible for doing is it basically cuts up protein. Now the first thing it does is it will cleave itself out of this long viral polyprotein. So we have it here cut away. And what it will do is it will go throughout the polyprotein, and it will cleave these other sites so that the polyprotein becomes its mature protein form. Now this is absolutely necessary for viral replication to occur. So what I've been doing is engineering this viral protease so that when I transfect a human cell with the protease, I give it information so that it makes this protease protein, but I attach it to a second protein. And in the presence of an antiviral inhibitor, you would expect that protease would do its job and cut itself out of this protein. However, in the presence of an antiviral compound, this cleavage event doesn't occur, so we can draw this over here. This protein will travel to the nucleus, and these cells are engineered in a specific way so that if this cleavage event does not occur, it will initiate a signaling cascade so that the cell starts to produce green fluorescent proteins, and we can visualize this using microscopy and flow cytometry. So what I do is in a tissue culture room, I will have small plates with anywhere from 12 to 96 individual wells, and in each well I can place human cells. And with each one of these populations, I can transfect these cells with the protease protein in the presence of different antiviral compounds. Now, when I go to visualize this plate, if I see that in, let's say, in this specific well, I see these cells are fluorescing green, that'll tell me whatever antiviral compound I used in that specific well is a potential successful drug. And what we can do is we could go on with further experimentations to make sure that, number one, it's specific for the virus that we're using, and number two, that it doesn't have any cytotoxic effects. If it meets both of these criteria, we can give this compound to a medicinal chemistry lab and hopefully make it more potent. And in the future, this assay that I'm developing will hopefully be used to discover a new antiviral drug for Zika. And other than that, it's just really important that I hope I end up finding something that can be used to help a lot of people around the world. Thank you.